Hello, welcome to chapter 12. It's a beautiful day today. I was sitting outside. I thought about giving my lecture outside, but there was a magpie that thought it was king of the neighborhood and was squawking every 2 seconds. And then my neighbor decided to mow the lawn. So we're inside, but facing the window. Today, we're going to talk about patterns of inheritance, Mendelian ratios, P's, Mendel's P's, and all that. Here in this picture, we've got some kids. You can see we've got kids with red hair, with brown hair, variety of different skin colors, eye colors, heights probably. So we're going to talk about how it is that we've got all these different traits, even amongst siblings, right? You can have variety. There are 6 objectives for this chapter, and this week's mini exam is going to cover this and the following chapter on Thursday. I forget what chapter that is, but so first of all, we're going to learn about Mendel's research system, his setup, why it was such a great setup. You should be able to explain a few reasons why it was such a good system. Next, you should be able to use Mendelian ratios, or to determine Mendelian ratios using a monohybrid cross, and then be able to do a dihybrid cross. And also at this point, you'll learn what independent assortment means, and also how you can see that being demonstrated in a dihybrid cross. Fourth, you should be able to predict the probability of a certain outcome. So we'll do a little bit of math. And then to understand what a test cross is and how you can use that to find out the genotype of an individual. Lastly, we're going to talk about some exceptions to Mendelian inheritance. He discovered a whole lot, but not everything. So first, let's talk about Mendel. So Gregor Mendel was a monk. He gardened pea plants and studied genetics and came up with just a lot of really amazing knowledge. And it's really great that he chose this plant because it was particularly well suited for studying inheritance. So first of all, pea plants, you can have hybrids of pea plants. Also, there are many pure varieties. So back then they called them pure. Because they didn't even know chromosomes were at this time, right? So what we call a homozygous strain, they would call pure. Right? So they had a lot of different homozygous traits, peas with homozygous traits that they could cross and see what happens. They're easy to grow, right? Pretty small. They have short generation time, so you can get multiple generations of plants over relatively short time compared, say, to studying in, say, a trait in dogs. And you can also do self-fertilization. So if you wanted to in-cross with self, we'll talk about how you could do that. And they can easily be cross-fertilized, so you can control what is fertilized by what, right? So let's go over a little bit about plant biology. Here we've got a flower on a pea plant, and what I want you to notice is the male and the female portions. So within this plant, the anthers are considered the male portion of the plant. Those are what release the pollen. And then the carpel is considered the female part of the plant, and that contains the stigma and the style. Right? So pollen lands on the stigma and makes this tube that goes down the style when this plant gets fertilized. So you can see how easy it is for this plant to self-fertilize. That pollen is right there, really close to the stigma. And so in order to prevent self-fertilization, scientists would just cut off the anthers. So they remove the male portion where the pollen is from that plant, and now it's much easier to cross-fertilize. And to cross-fertilize, all scientists have to do is take a brush to pick up some pollen from one flower and then put it onto the stigma of the other flower that they want to fertilize. Then they take those resulting pea plants and plant those resulting peas, plant them, and look to see what the properties are of the plants that grow from those individual peas. So in order to control what is fertilized with what, scientists will also do things like put bags over plants. That way, say once, if they don't want something to get fertilized, or if they have already fertilized it and they don't want any contamination, that's a great way to prevent pollen from other plants getting onto your flowers. So there are three main stages of Mendel's experimental method. First, he would get some true breeding strains for each trait. And so again, they didn't know what chromosomes were back then, homozygous or heterozygous. And so what we consider homozygous is called true breeding. So homozygous dominant plant or homozygous recessive plant. And then they would cross-fertilize these true breeding strains. So for example, say you have a plant that one plant that is homozygous dominant that results in yellow seeds, and another plant that is homozygous recessive that ends up with green seeds. Cross those two homozygous strains or true breeding strains and look to see what color are the peas in the offspring. Then they will also, after they have that first generation, in-cross or self-fertilize that first generation and look to see what happens in that second generation in terms of the traits. So let's look at this in an example. All right, let's look at a monohybrid cross. So we're talking about traits, right? So usually when you're talking about traits, in this class, what we're going to talk about is just having two variations. So if you're looking at pea pod color, green or yellow, there's just two options. Or flower color, purple or white. Pea shape, wrinkled or smooth, right? Just two variations. And so he had 7 different traits, which had 2 variations each. And he had these true breeding strains that he could use and cross. So here's some of those examples that I was talking about. We've got seed texture, color, flower color, position of the flower, shape of the pod, right? You can see for each of these traits, there are 2 variants only. So again, we talked about how he had kind of these 3 steps. The first step was to get true breeding plants and to cross them. So here we've got, in this example, we've got a true breeding or homozygous dominant and a homozygous recessive plant. And the one trait that we're looking at is flower color. And there's 2 variants. The flower color could be purple or it could be white. Now, we know the genetics. We're talking about chromosomes and alleles. We're talking about the purple plant has 2 dominant alleles. It's homozygous dominant. And the white plant is homozygous recessive with those recessive alleles. And so for this, we're going to demonstrate the dominant trait as capital P and the recessive trait as lowercase p. And so when you cross these 2 homozygous or true breeding strains, you get all heterozygous plants, right? So what color would these all be if purple is the dominant allele? They would all be purple. Okay? So for every trait he found, this happened, whether it was flower color or pea pod shape. When he would cross 2 true breeding or homozygous strains, all of the offspring would look the same. Right? And so that was how he was able to discover this property of dominant and recessive traits. He was the one that showed that. And so we know in this example, because all of the offspring are purple, that tells us that the white flower color is the recessive trait. If, for example, we cross these 2 plants and they all looked white, that would tell us that white was the dominant trait, right? An important note here, we don't see any intermediate characteristics. We don't see any like light purple flowers, right? It's either purple or white. There's no intermediates mixing. So let's look, now that we know how meiosis works, let's look at this in detail kind of from the perspective of what's happening with these germ cells. So the purple plant and the white plant, they are homozygous, right? The purple plant is homozygous dominant and the white plant is homozygous recessive. And the germ cells in this plant start out diploid, right, and undergo meiosis to provide gametes, these haploid gametes. So what are all the possible combinations of gametes that we could get? For the purple plant, all of the gametes that result from meiosis are going to just have that dominant allele, right? That's the only option to produce. And in the white plant, all of the resulting gametes are going to be recessive, have the recessive allele. And remember, gametes are haploid. They only have one copy of the gene. So we know that these are all the possible gametes that these plants are going to be producing, these individuals are going to be producing. And so when fertilization occurs, you're going to have, right, egg and sperm, one male and one female gamete are going to fuse to form a zygote. So there's really only one combination that you're going to see here. The only possible combination is the heterozygous offspring. And you can see how this fertilized embryo is now diploid because we had two haploid gametes fuse. And that is exactly what we see when we do our Punnett square, right? You can see that when you cross a homozygous dominant and a homozygous recessive, that all of the offspring are heterozygous. So that was the first part of Mendel's experimental method. The next thing that he would do is he would do an incross, right? So he would take that first generation and self-fertilize, allow them to interbreed. And so the resulting offspring of that are the F2 or second filial generation. So on the right you can see we've got the results of the first mating. We had all heterozygous plants. And so allowing them to inbreed, we're taking two heterozygous purple flowers and crossing them. And you can see that the result of this is that we have three purple flowers, or three chances for purple flowers and one chance of getting white flowers, that recessive, homozygous recessive. And so he did this over and over with all sorts of different traits, and he always found this 3-to-1 ratio. When he would take the second generation and inbreed them, there was this hidden trait that would be revealed in only a quarter of the offspring. So again, let's look at this in perspective of meiosis. So we have two plants that are heterozygous that we're crossing. Their diploid germ cells are heterozygous, and the resulting gametes of meiosis of each of these plants, we've got two possible results. They could contribute either the dominant allele or the recessive allele. Now, when these gametes fuse during fertilization, we have a few possibilities, a variety of different combinations that can occur. You can see that 1/4 of them are going to be homozygous dominant, half of them are going to be heterozygous, and 1/4 is going to be homozygous recessive. And again, that is just like what we see in our Punnett square, right? It's just all the different possible combinations of gametes from those two parents. Here we have some data. You can look at the F2 generation. So when they crossed the offspring of that first mating, they found, for example, in flowers, that we've got 705 purple flowers plants versus 224 white flowered plants. That ratio is 3.15 to 1, so it's about 3 to 1, like we said on the previous slide. And he did this over and over again with different types of traits such as seed color, texture, pod shape. And if you look at these numbers, 3.01 to 1, 2.96 to 1, 2.82 to 1, they're all roughly 3 to 1. And so that was what allowed him to really understand and tease apart this dominant and recessive trait and this idea of heterozygotes, right? Now, when we say 3 to 1, I just want to take a minute. We're talking about the phenotype, the color, right? 3/4 of the offspring are purple, 1/4 is white, right? But when you're talking about genotype, it's actually 1 to 2 to 1, right? One of the 4 offspring is homozygous dominant, 2 of them are heterozygous, and 1 of them is homozygous recessive. So the appearance phenotype visually is 3 to 1, but the genotype is actually 1 to 2 to 1. Right? And he didn't use the word heterozygous, he called them not true breeding plants. Because again, they didn't know about chromosomes back then. So here's a nice overview of his system, right? So he starts out again with 2 homozygous strains, 2 variants of this gene, purple or white for flower color, cross-fertilizes them, and he gets the resulting F1 generation. And so the trait that he gets, that helps him determine which one is the dominant trait and which one is the recessive trait. In this case, because all of the offspring are purple, that tells us the purple allele is the dominant allele and white is the recessive allele. And he calls this non-true breeding, or heterozygous offspring, because when he allows them to self-cross, he does not get all purple offspring, right? He gets 1/4 of them are white that are homozygous recessive. And then he can further prove that that some of these are actually heterozygous by allowing them to self-cross again and reveal that hidden trait. Whereas the one plant that is homozygous dominant, when you self-cross it, all of the offspring are purple. Same with the white recessive homozygous flower plant. When you self-cross it, you're only going to get white. Again, some of the things that he really was able to prove was that for each pair of alleles for a certain gene, there's going to be one trait that's dominant, another is going to be recessive. You don't show intermediate or mixed traits. They're discrete. It's purple or white, green or yellow, wrinkled or not. You don't get kind of wrinkled or yellowish green, right? Also, let's see, the alternative traits, or these heterozygous plants, were revealed or demonstrated in the 2nd generation. When they incross the 1st generation, they show that 1/4 of those are recessive. We've got that 3 to 1 ratio. So some of this might be a little bit of review, but I just want to make sure we're all on the same page. So there's this 5-element model that is used when we're talking about Mendel's work or Mendelian genetics. Again, first, parents transmit discrete factors, these genes. Each individual receives 1 copy of a gene from each parent. And not all copies are identical, right? These variants of a gene are called alleles. And when you have 2 of the same allele, it's called homozygous. And when an individual has 1 of each allele, it's considered heterozygous. Fourth, these alleles remain discrete. You don't have mixed or blending of traits. And the presence of an allele does not guarantee expression. So that's the idea that if you are a heterozygote, the dominant trait is going to win out, and the recessive trait will be hidden. When we talk about genotype, we're talking about what specific alleles does that individual have. And when we're talking about phenotype, we mean what is the physical appearance, right? Because something can have a heterozygous genotype, but have a dominant phenotype, right? So an important principle that Mendel revealed was the principle of segregation. And some of these may seem kind of like, well, duh, but they had to be proven, right? So the principle of segregation is that when you have a fertilized embryo, that it is diploid, it's received one copy of the gene from one parent and one from the other parent, right? Basically, only one of the two gene copies that are present in an organism is distributed to each gamete. So in this picture, we've got two individuals, right? And their germ cells, when they create gametes, those gametes are haploid, right? The germ cell, for example, has two copies of this gene A, but in the resulting gametes, it's only going to have one. Okay? One of the two gene copies in an individual is distributed into each of its gametes that it makes. Also, this is random, right? Again, he had no idea, he hadn't heard about chromosomes, so it's pretty neat that he was able to figure this out. So let's talk a little bit about human genetics. So we can actually track traits in people in family histories, and you can determine whether traits are dominant or recessive by tracking the inheritance patterns. This is called pedigree analysis. And one example of a dominant pedigree is the disease juvenile glaucoma. This disease affects the optic nerve and can impair vision. And this is the gene, the mutation that causes this juvenile glaucoma. The gene, the allele that has it is the dominant allele. And so let's look at this example pedigree, a dominant pedigree. So in this case, we have generation 1. The father has the disease allele, the mother does not. And in the offspring, we can see that some of the children have it, some of the children don't, right? And we can see that basically in every single generation, somebody is affected by this disease because it's a dominant trait. If you are heterozygous, you are going to have the disease, right? So in this case, the fact that in the second generation that some of the children had it and some of it didn't, that indicates that the father was heterozygous for the disease because he was able to pass on the healthy allele to some of his children, but not all. So let's look at some dominant and recessive traits in humans. So having hair on the middle segments of fingers apparently is a dominant trait. Having short fingers, mine are kind of stubby. I don't know if I qualify for brachydactyly, but other things, let's see. Elevated levels of blood cholesterol and heart attack. Polydactyly, having an extra finger. These are all examples of dominant traits. Recessive traits include albinism, can't talk today, red-green color blindness, cystic fibrosis. So in these recessive traits, you're going to have people that have the allele, the diseased allele, but don't exhibit the disease, right? They are considered carriers. And so that is what you're going to see quite different in the pedigree. So again, in the dominant pedigree, you saw the disease show up in every single generation. And you'll see that it's not the same with a recessive pedigree. Let's look at the example for albinism. In this, there's a gene which codes for an enzyme tyrosinase. It's important for developing pigment. And the mutated form prevents that enzyme from functioning. And so individuals that have two copies that are homozygous recessive will have no pigmentation. Those that are carriers, so those that are heterozygous or that are homozygous dominant, will not have albinism. Now, when you look at this pedigree, you can see something different. So in generation 2, we've got some examples of individuals that are half green and half white. What that means is that they are heterozygous. In this case, because the allele is a recessive allele, will those individuals have albinism? No, they will not, right? So this entire second generation does not have albinism. And in that first generation, none of them had albinism. However, somehow in the third generation, we're seeing individuals with albinism. And so you can see how much less organized and predictable this pedigree is. And that is one of the indicators that it's a recessive allele. So in generation 1, one of these parents, they don't know who, based on this history that they got, one of them was heterozygous. Again, it's a recessive allele for the disease, and so if you're heterozygous, you won't exhibit, you won't have the disease. So the parents appeared to have healthy alleles, but in fact, one of them was hiding the diseased allele in their genes. And then in the offspring, all of the offspring had the healthy allele. However, some of them were carriers. And so when a carrier, such as individual 4 in generation 2, married, I assume, an individual that was homozygous recessive, that didn't have the disease, they ended up having one child that was also a carrier and 2 that didn't have it at all. In this other example, 2 and 3, you have 2 heterozygous individuals, don't have albinism. Their children, half of them actually did have albinism. They lucked out and they were homozygous recessive for the allele. And so you can see, right, again, you can have this people be carriers of these traits, and this trait will pop in and out of the family tree when it is a recessive pedigree. Let's talk about dihybrid crosses and independent assortment. So the idea of independent assortment is a little bit confusing at first. And so I want to go over an example first, and then you may kind of go back to this slide and it may make a little bit more sense about what I'm talking about here, right? So first of all, what is a dihybrid cross? So a dihybrid cross is when you are involving more than one trait. So in the past, we just looked at one trait, flower color. It could be purple or white. What if you were crossing plants in which you were looking at flower color and seed color? So now you have 2 traits or 2 genes that you're tracking. And so that's considered a dihybrid cross. And so these alleles get sorted into gametes independently of one another, right? The allele that it gets for one trait doesn't affect the allele that the gamete gets for another gene. For example, just because an egg has the allele for white flower has no impact on whether or not that gamete is going to get the allele for green or yellow seed color. Let's look at this in a real example. So again, we're examining 2 separate traits in a cross. So in this example, we're looking at seed color and texture. So we have seed color, yellow or green are the options, and yellow is the dominant trait, capital Y. Green is the recessive trait, lowercase y. And then we have rough, round or smooth. Round is capital R, or sorry, round or wrinkled. Round is capital R and wrinkled is lowercase r, right? So we've got a homozygous plant that's homozygous dominant for color, it's yellow, and it's homozygous dominant for texture, so it's smooth, and we're crossing it with a homozygous recessive plant for both of these traits, so it is rough and it is wrinkled and it is green. Now when these plants are crossed, the F1 generation, the resulting generation, just like before, is heterozygous, and it's heterozygous for each of these traits. Then the scientists will perform an in-cross of that F1 generation, and we have the following, we're going to get a variety of different combinations in the F2 generation that I'm not quite showing yet, right? So here we have, let's look at this cross in terms of meiosis, right? What are these gametes carrying? So when each of these individuals undergoes meiosis to produce haploid gametes, again, we don't see a lot of variety, right? If it's homozygous dominant for both traits, it's going to only produce gametes that are dominant for those traits. And same for the recessive, right? It only has recessive alleles, and so that's all that it can contribute to the gametes. So these gametes, again, they're haploid. They only have one copy of each gene, one letter R, one letter Y. And when these two individuals, when they mate, the potential combinations here, you can see they're all heterozygous for both genes. Right, so this is just really breaking down that first generation. Now, the next part, right, we have to in-cross that first generation to produce the F2 generation. So we have our diploid germ cells that are heterozygous. And when these germ cells undergo meiosis, here we have all the possible gametes that could be created. So here you can see there's more variety, right? We can have some gametes that have the dominant allele and some gametes that have the recessive alleles. So we have all these potential combinations, right? And you get this massive Punnett square. I encourage you to practice drawing one out on your own just to really cement this concept, right? And so let's look at some of the combinations that we can get. Again, we're taking one gamete or one egg and one sperm, fusing them, and that result is in the center of our Punnett square. And so when you add up all the different phenotypes, you can see that 9 of these are going to be round and yellow, 3 of them are going to be round and green, 3 of them are going to be wrinkled and yellow, and only 1 is going to be wrinkled and green. Right? So here we have a 9:3 to 3:1 ratio. Right? So how does this relate to the law of independent assortment? So you've learned how to do a dihybrid cross, so great. Again, this is what happens for formation of the F2 generation. When the F1 generation undergoes meiosis, we've got all these different combinations of alleles that can be put together. So why is it that when meiosis forms that the individual, the gametes, that one of them is capital R, capital Y, one of them is lowercase r, lowercase y, another is capital R and lowercase y, and then the last combination potential is lowercase r and capital Y. Why do we get 4 potential outcomes? Why don't we get, for example, something like this? When meiosis occurs, why is it that we get 4 options and not, for example, 2? Right? This is because of the law of independent assortment. We are just assuming that these gametes, when they're formed, that the alleles are going to separate out into the different gametes independently of each other. It's not like if you have a capital R, that means you're also going to have a capital Y. If you have one allele for one gene, it does not influence what the allele for the second gene is going to be. Let's look at this in another diagram. Here we have two individuals looking at their germ cells, and during meiosis, the gametes are going to be formed that are haploid. All right. So one copy of the gene of the alleles for the R gene is going to go into the gamete, and one copy of the two alleles for the Y gene is going to go into that gamete. Right? And they aren't connected in any way. All right. Now, this is a tough concept. This video really helps you to understand the rule of independent assortment. So if you want to do well on this concept on the mini exam, I encourage you to take a minute to watch this YouTube video and then go back through these slides and just look at them again, and it'll make a little bit more sense if you're struggling. All right, so let's talk about probability. So probability allows us, using these rules, math rules, we can determine the likelihood of different traits appearing in offspring. So the first rule we're going to talk about is the rule of multiplication. Right? This is the probability of two independent events both occurring. To determine that, it was the product of their individual probabilities. You're thinking, oh my gosh, this is so boring, right? So let's look at an actual example. So when crossing two heterozygotes, say for flower color, what is the probability of obtaining homozygous recessive offspring? So here we have our little Punnett square. Now we don't have to do a Punnett square every time. We can use these rules, these math rules, to figure out the probability. So in this case, to have homozygous recessive offspring, during fertilization, the egg has to have the recessive allele and the sperm has to have the recessive allele, right? We're looking at an and situation. We have to have both of these, right? And so in this case, whenever you're thinking of and, you're going to use the rule of multiplication. So what is the probability or the chance that the mother is going to contribute a gamete that has the recessive allele? There's only two options, right? Dominant or recessive, so it's 1/2. And the chance that the father will provide the recessive allele as well is 1/2. So you're going to take 1/2 times 1/2, and that's going to give you 1/4. So there is a 1 in 4 chance that the offspring of this cross will be homozygous recessive. And if you were to do your Punnett square, you can look and see that that is correct, 1 out of 4 chances. Now let's look at the rule of addition, right? This is when you're looking at an or situation. For example, to have flowers that are purple, they could have two different genotypes. They could be homozygous, dominant, or they could be heterozygous, right? And so we actually have these 3 potential outcomes, right? The dad could contribute the dad could contribute the dominant allele, and the mom could contribute the recessive allele to heterozygotes, or vice versa. You could have the dad provide the recessive allele and the mom provide the dominant allele. Or you could have both mom and dad providing the dominant allele. And so for each of these, we know that there is a 1 in 4 chance of them happening. And so because we're looking at this or situation, we're going to add up these probabilities. And so 1/4 possibility of getting this heterozygous dominant, where the dominant allele is from, say, the dad, and then 1/4 in which the dominant allele is from the mom instead, and then 1/4 of the offspring are homozygous dominant, and when you add those all up, you get 3/4. And again, if you were to write out your Punnett squares, we see on the right, that is what we find. 3 out of the 4 potential combinations are purple. Okay. Now let's talk about what a test cross is and how to use it. So we talked about how you can have these people or these individuals that are carriers, right? But how do you know who is a carrier and who isn't? And so you can't do this in people necessarily. You can look at histories, but in plants it's quite easy. You can perform a test cross. And so you can say, all right, we don't know if you are homozygous dominant or if you are heterozygous. Let's cross you with something and see what your offspring look like, and that will reveal to us what you are. Right? And so you're going to take the individual with the unknown genotype and you're going to cross it with a homozygous recessive. Right? Then you're going to look at the ratios of the offspring phenotypes. In this example, we've been using the flower color, so let's just stick with it. Right? We have a purple flower. Is it homozygous dominant or is it heterozygous? So to answer this question, we will perform a test cross, and all that means is to cross that individual with a homozygous recessive individual. So in this case, it would be crossing our purple flower that we don't know if it's homozygous or heterozygous with a flower in which we do know that it is homozygous recessive, so a white flower. Now, we have two possible outcomes, right? If the plant is homozygous dominant, when it is crossed with a homozygous recessive, all the offspring will be purple and they will all be heterozygotes. But if the parent, this unknown individual purple, is heterozygous, when you cross it with a homozygous recessive flower, you are going to get half of them be purple and half of them be white. See how we've revealed that hidden trait? And so by looking at the offspring and what percentage of traits you have, then you can determine what that parent, if you can call plants parents, what it is. Right? So you should be able to know how to do a test cross. If you were given the outcomes of a test cross, you should be able to say, oh, this parent was homozygous or it was heterozygous. Lastly, we're going to talk about, this is the last objective, some of these exceptions or additions to Mendel's rules. Again, what does he assume? He assumes that each trait is controlled by a single gene, that there are only two variations for that gene, green or yellow, purple or white, and that there's a very clear dominant recessive relationship. That is, if you are heterozygous, the dominant trait will always win out. If you are recessive, the only time you're going to see that trait is if it's a homozygous recessive individual. However, most genes actually don't meet these criteria, and so Mendel was very lucky in that he chose peas to do his research, because when you look at other types of species, it gets to be a little more complicated. So we have something called polygenic inheritance, and we've talked about this before. You can have a trait that is controlled by multiple genes. So an example of this is human height, right? We do not have two heights that everyone is, right? It's not like you are either 6 feet tall or 5 feet tall. That would be horrible, right? You can be 5 feet tall, you can be 5'2", you can be 5'8", you can be 6'3". We have this range, right? And we have a normal distribution. So this is a really cute histogram that they made with their heights. They organized themselves, right? On average, we can see in this group, the average height is 5'8", but we have a range, right? This normal distribution, a bell-shaped curve. And that is because we have multiple genes that are contributing. It's not like we have one gene that says you are tall or you are short. Another exception to Mendel's rules is pleiotropy, and that is where one gene can have a variety of effects on an individual. So one example is cystic fibrosis. This is caused by a mutation on one gene, right? So you could have the mutant allele or the normal allele, healthy allele. And in the mutant, it's not like they just have one thing that is wrong, right? There are a variety of things throughout that individual's body, that organs and cells that are not functioning properly. Multiple symptoms or multiple traits traced back to one allele. We also talked about with sickle cell anemia, how individuals that are heterozygotes, they also have resistance to malaria, right? So again, they've got two different traits resulting from one single mutation in one gene. We've always talked about having two alleles for a gene. You are purple or white, right? There's really no limit to the number of alleles that there can be for a gene. Anytime there's a mutation in a gene, there's a creation of a new allele. Think of how many bases there are in a gene, right? So one example in nature, right, are blood types. For the blood type gene, there are 3 alleles. So you could have A, B, or O. However, we're still diploid. We're only going to have 2 alleles. So you could be AB or AO, right, or AA or BB, right? So this is an example, right, where there are more than 2 alleles. So how do you draw that Punnett square, right? Another thing, we've talked about these rules of dominant, this rule of dominance, that if you're heterozygote, the dominant allele will win out all the time. Unfortunately, that's not always true. It was in the pea plant, but not in humans, right? We talked about how people that have the mutation in the sickle cell gene, how they will result in these people, it's a recessive trait, right? So people that are heterozygous for the sickle cell allele, they don't have the disease. However, they still have some beneficial, type of resistance to malaria, right? And this is because when they get infected with malaria, their blood will change. It will become sickle cell-like. And so what that's telling us is that we have incomplete dominance, that heterozygote does not always act like it's dominant, and the recessive allele doesn't always act recessive. So there are some cases, for example, where you can have a red allele and a white allele for flower color, and one of the possible combinations could be pink, right? So just breaking the rules all over. Another example is codominance. So in this case, the heterozygote shows some aspect of both traits, both alleles, such as type AB blood. It's not like there's a dominant allele that wins out over the other. One of my favorite exceptions to Mendelian genetics is the role of environment. This is a pretty cute example in coat color in Himalayan rabbits and Siamese rats. So there's a gene that produces a pigmentation enzyme. This enzyme is important for production of dark pigment, and this enzyme is only functioning in cool temperatures, so below 30 degrees Celsius. So what you'll find is that in the body of this individual, this little cat, that only the parts of the body that were able to get below 30 degrees, only those parts in those cells was the enzyme able to function. And so we've got pigment in the ears, the tail, the face, parts of the body that are cooler. However, in other parts of the body that are warmer, even though they had the allele for pigment, the enzyme didn't get warm enough, and so they didn't have pigment in that fur. The last exception that I want to talk about is called epistasis. And this is when you have two genes that are affecting each other, right? So, for example, you could have two genes, one that's responsible for fur color, such as brown or black, and then you can have another gene that's just responsible for whether or not there's pigment, so pigmented or non-pigmented. And so you could have an individual that, say, is homozygous black, all right, for the fur color gene, but it is homozygous non-pigmented. And so in this case, that animal is going to be white, it's not going to produce pigment, and so it doesn't matter whether it was brown, got the gene allele for brown or black. It doesn't even have the ability to form pigment, right? Another example of this is having a gene for baldness and a gene for hair color, right? It doesn't matter what gene you inherited for hair color if you have interruption by this other gene that doesn't even allow you to produce hair, right? So that is it for chapter 12. I just want to say good job to everyone for hanging in there. We're almost done with the semester. Please do remember to email me with any questions. And I thank you for your patience, as I know I've been learning about recording videos and keeping things up to date on Canvas. Y'all have a good day.